Hey, Assalamu alaikum. In this video, we will understand about the hyperglycemia and polyol hexosamine pathway. So the sorbitol and hexosamine pathway. So the hyperglycemia mean the high level of the glucose in the blood, and polyol mean the basically the sorbitol increase. So that is pathway which that is dangerous for some cell, and some cell is beneficial like the gonads and ovary required it because of fructose production. So anyhow, later we will discuss. Here is the whenever you will intake the carbohydrate, so carbohydrate will convert into the simple sugar, enter into the blood circulatory system to target to the beta cell of the pancreas, and will release the insulin with the response of the hyperglycemia. This for normal person, the insulin release is known as the exocytosis. Will bind with a special cell which that contain insulin receptor to allow the glucose move inside. So the influx of the glucose. In this way, the glucose will increase. In this way, important thing you should need to understand whenever the glucose increase, so the insulin resistance will occur. So some cell, basically like endothelial cell, not require the insulin receptor, so directly increase the extra glucose, move into the endothelial cell and other cell, and in this basically the glucose when enter will convert into the sorbitol because the extra glucose not required in the cell, so sorbitol conversion. The sorbitol require the more water for the dilution, so in this the build up the osmotic damage will occur because the osmotic build up, so osmotic lysis will occur. In this the water build up in the cell extra will lead to burst the cell and necrosis will occur. So remember, the osmotic gradients will create due to the sorbitol production, and the sorbitol is more higher for absorption of the water as compared to the glucose. So in this the sorbitol for some reason, and the some cell require the sorbitol, but some cell every cell contain less sorbitol, not excessive because normal glucose. So here is the increase of glucose for the bigger picture to understand glycolysis to produce the glyceraldehyde 3 phosphate, and this glyceraldehyde 3 phosphate and increase due to the increase of the glucose which that convert into the pyruvate, and pyruvate increase and methyl glyoxal which that increase the advanced glycation end products, and the protein kinase C will activate due to the diacylglycerol production from the glycolysis, and in this way the glycolysis will increase, so the PKC, the protein kinase C will increase. On the other hand, the methyl glyoxal will increase and pyruvate increase. Pyruvate increase will enter into the mitochondria to increase more reactive oxygen species due to the excessive electron transport chain and electron leakage. The leakage of the electron will increase the reactive oxygen species and the oxygen superoxide and the hydrogen peroxide production. This will catalyze enzyme to convert again water and oxygen, but it is rare due to the more requirement of the superoxide dismutase and catalase enzyme, but the deficiency will lead to damage the DNA due to the increase the reactive oxygen species. And also the reactive oxygen species will lead to diabetic neuropathy, so discussed later in this video. So here is the glucose will convert through the aldose reductase enzyme to sorbitol formation. This sorbitol also requires the NADPH for oxidation of the NADPH to NADP plus. This is the cofactor. In this the glutathione, which that is the antioxidant, glutathione oxidized due to the NADPH deficiency, because the glutathione reduction required the NADPH, so the deficiency of the NADPH will decrease the glutathione. In this way the mitochondria will further damage due to the glutathione is very important for the prevention of the mitochondrial damage. So on the other hand, the superoxide sorbitol dehydrogenase enzyme also require the NADH, so the production of fructose, which that is important for the spermatocyte and ovary, but not for the endothelial cell. In this way, the blood vessel will be damaged. So the damage of the blood vessel will occur, and in this, on the other hand, the hexosamine pathway also occur, which that also damage the DNA due to the glucosamine 6 phosphate, which that convert into the by the increase of the glucosamine 6 phosphate This glucose 6 amine 6 phosphate will convert into the UDP, and N-acetyl glucosamine linked with the UDP to increase, and the O-linked glycosylation will occur. In this, the O-linked glucosamine and acetyl glucosamine will increase, and this will lead to damage the DNA due to the bond with the DNA, and this DNA will produce the mRNA, messenger RNA, to translation to synthesis certain type of the protein, which that is the cytokines and the inflammatory mediators. And the same inflammatory mediator will trigger due to the PKC increase due to the diacylglycerol due to the increase the glycolysis. So in this, the damaged DNA will produce the certain type of the protein P81, necrotic factor kappa B, and the transforming growth factor beta 1, and the vascular endothelial growth factor, which that increase, and same mechanism also increase due to the methylglyoxal 2-AGs produce diabetic neuropathy due to the advanced glycation end product. On the other hand, the protein kinase C activate to also activate the certain type of the inflammatory mediators. This inflammatory mediator will lead to damage the blood vessel to cause the diabetic neuropathy, because the nerve not get the nourishment from the blood.